We're now going to leave GoEnrichment behind, and we're going to move on for the rest of the lecture working on gene set enrichment analysis. So thinking back to the slides, this is the method that I used the card counting analogy to describe. So then it should follow that what we need for this gene set enrichment analysis is all of our data, right? Because that gene expression data is going to be used to rank order our list. And it could be gene expression data. It could also be like log fold change between two or more conditions. I need some numeric data so that the entire data set can be rank ordered. And then that data can be thrown away and that GSEA process can begin. Each individual signature or entire collections of signatures can be used for either self-contained or competitive GSEA analysis. All right. So one thing we need then to get started, and we're now going to do GSEA using Cluster Profiler, is we need the actual signatures, the collections of signatures. So the comments I have here really point you to several different ways we can access molecular signatures database collections or MSigDB collections. So the first option is you can directly download MSigDB collections to your computer and load them into your R environment. So to just illustrate what I mean here is we can actually go to the GSEA, the Broad Institute's website. Remember, they initially developed the GSEA algorithm and then developed the MSigDB right here to complement it. We talked about these collections in the lecture, but you can go to the download page and you can download entire collections. Like this is all of C2. You would just click here and it would download. And you can do that for any of the collections, or you can get any subset of the collections, and they're broken down a couple different ways. For example, under C2, you could get just the BioCarta signaling pathways or just the KEGG metabolic pathways. Each of these is downloadable as a collection. So it's a useful way to kind of think of how you might want to break up or approach your functional enrichment analysis using GSEA. And it's not always better to query more signatures, right? There's a reason why we're not always just looking at every possible gene set. That's more multiple testing correction that has to be done, and your p-values will take a harder hit. So if you think you're interested in metabolic pathways, why not just directly run gene set enrichment analysis using just C2 canonical pathways KEGG. So C2, CP, KEGG, all right? So you could download these and then put wherever they are on your computer, point the function read.gmt from the cluster profiler package, point it to the file path that it can find that GMT file and read it into your R environment. I tend to read in the symbols files, not the entree IDs, because I usually have gene symbols as identifiers. Remember that we're going to be looking for these signatures in our ranked data, so the IDs need to match. So I have symbols in my data, so I'm going to download the symbols file. You'll find entree ID files for each symbol file as well if you prefer that. All right, once I do this, I've read that collection, C2, CP. I didn't go down to KEGG level. I just took all the canonical pathways from C2, and I read that into an object called C2CP. I can see that that's over 100,000 observations of two variables. So if I click on that, what I can immediately see is this appears to be a data frame, and a tidy one at that. So Hadley Wickham would be happy. We have two variables: the term, which is the name of the signature, and gene. And in this case, this is all the genes, each row, each observation, that belong to the KEGG glycolysis gluconeogenesis pathway. And I can see there's a number of genes that belong to that pathway. As I get lower down in this, there's over 60 genes that belong to that pathway. As I get further down, now I'm at the citrate cycle, TCA cycle, KEGG pathway, and it has its respective members and so on. And this continues on for 100,000 plus rows. So that's one way to get data in. But the challenge or issue a bit with MSigDB is it's very human oriented, and there's been no tailoring of these collections to account for the fact that there are expansions or contractions in gene families between species. So to make a species-specific MSigDB possible, several groups have undertaken the manual work of making these species-specific versions of MSigDB. And fortunately for you, they're now available through an R package, and that's the msigdbr package. So this is option one. Option two is to use the R package msigdbr. You can see what species are available using the msigdbr show species function and not passing any arguments to it. So I can just run, and I can see that there's 11 different species available and a decent variety. So now I can run msigdbr and choose human, and I have to type it exactly as it's written here. When I do that, the object that I'll return, I'm going to go ahead and start it now because it might take a few seconds. What I have accomplished here is I have read into the R environment every single collection from msigdb for human. So I have every single signature in the msigdb database, and I have human gene symbols for it. I can see that the object that I produce, if I look at HSGSEA, is not 100,000 rows long. It's 3.3 million observations of 9 variables. That's a lot, so I'm not going to try to open that right now, but if I now just pass this object, right, this data frame, to dplyr, I can start to work with it because it's a big data frame, right? And there's a column header in this data frame called gene set category and another one called gene set subcategory. So this is really the collection and the sub-collection. So I want to ask, I can use the distinct function from dplyr to only show basically the values, the factorized values of those variables, and then arrange them in order. So when I do that, it will just display, because I haven't saved it to anything here, just display in the terminal, in the console, and I can see that now that took that 3 million long thing and reduced it to a distinct arranged data frame where I can see there's collection C1, C2 CGP, chemical and genetic perturbations, C2 CP, canonical pathways, C2 CP biocarta, KEG. So you can see all the collections and subcollections for each of these sort of areas of the msigdb, and H is the hallmark collection. All right, now that I see the layout of this, and remember, I didn't modify HSGSEA, I just passed it through these dplyr functions to produce this in the console so I could just see it. But now that I know what I'm looking at, I can now go back and do what we really would have done from the beginning, which is pick a species and a category. So I'm going to choose human C2, all of C2, and I just want to pick the gene set name and gene set symbol columns. And I do want to save what I produce from this to a new object, not called HSGSEA, but called HSGSEA C2 for the second collection C2. I run this, and now I've produced this HSGSEA C2. If I open that in my viewer, it looks exactly like what I read in using the first option. It's a tidy table that I can see the association between each gene symbol and its respective signature or gene set. All right, now that we have our msigdb collections ready, we need our data. For this, you have to think back to that step 3 script where we had this mydata.df, and I will, actually we can just run it from here because I think it's a Tibble. Mydata.df is really, you should absolutely recall this, it's just all of our data, but it's after we took dplyr to it a little bit and added the average columns at the end, and then the fold change, the log fold change as the last column. So for the purposes of carrying out the GSEA, I need some data, but I don't need all this. So I just want to take that data frame. I want to select from that data frame the gene ID, because we need IDs to carry out GSEA, and I want the log fold change column. That will suffice for giving me a rank order of my data. And I'll put that into an object called mydata.df.sub. Right. So we now have that simple structure here. Now I'm ready to do a few things to get this into a shape that I can actually use it for GSEA. The first is I'm going to turn it into a named vector. In other words, I want each of these values to have the name of the symbol associated with it. So I'm going to pull out the log fold change column, and put it into an object called mydata.gsea. That is a numeric vector. I'm then going to take the gene ID column as a character vector and put it into the names of that numeric vector. And once I've done that, if I look at now the object I have a named vector, and I can now sort that named vector in decreasing order. That's it. We now have an object that is exactly the kind and structure that we would need for GSEA. We have rank ordered our data. Now that we have that, we're ready to run GSEA, at least using this first method we'll try, which is the GSEA function from Cluster Profiler. It will use the approach that I described earlier. It will permute gene labels, not sample labels. So it is subject to these intergene correlations that I described. So now that we have that, we have our data, we have our collection of signatures, we are ready to run GSEA. So it's running against a lot of different signatures. I can't remember how many are in C2. I think it's on the order of several thousand different signatures just in C2. It may actually have it on the website. Used to have it written here below each one, but I don't see it anymore. It doesn't matter. It's taking each of those signatures and it's doing what I described in the slides. It's taking the rank ordered data, and it's looking through that ranked data for each member, each gene member in that signature, and it's working one signature at a time, and it's generating a running sum statistic, and then it's permuting gene labels, and it's redoing that analysis to generate a distribution of enrichment values. And from that distribution and the actual observed enrichment value, it can generate a p-value. All right, so this will be done in just a second. Once it's done, we need to extract from the result the data in the form of a tibble. Okay. Now, one thing while this is finishing, you may notice that this GSEA result, we're accessing it not using a $sign, but using an @ symbol. And I think this is the first time we've seen this pop up in our scripts. This is just a way to access a slot. You can think of this as an element in a list, if you will, to access a slot in a type of object or a class of object in R called an S4 class. We're not going to get into S3 and S4 objects, but it operates for all intents and purposes for this point in our course. Let's just think of it as operating in a manner similar to the $sign, but instead of on data frames or lists, it's operating on an S4 class object. So it's pulling out one piece of that object. So I have the GSEA result. It's done. It listed a warning here, which is fine. I'm now going to extract the result portion of that as a tibble, and I'm going to store that in something called mygsea.df. All right. I have this data frame. It's a tibble, so it'll display nicely here, that now has each pathway named, the size or number of genes that are in these pathways. Has nothing to do with my data. This is the number of genes in the pathway in MSigDB. I get an enrichment score, a normalized enrichment score that's been adjusted for the size of the gene set, a p-value, an adjusted p-value, q-values, and the rank. All right. Now that I have that, I can just use the DT function, DT package data table function like we've used before, and I can go ahead and make an interactive table that's completely searchable. There. Now I could search for any gene, any signature I want. If I'm interested in CD8 T cells, I could search for those, or cytotoxic. There we go. I can see, as something we've observed in this data before, that these cytotoxic T cell pathways are highly enriched and highly significant in the skin of patients with this disease. All right. So you can now produce this interactive table, but many of you are familiar with these enrichment plots, and in fact I showed you one in the slides, this idea of actually plotting the running sum statistic. So let's plot that for one of these. Here is KEGG natural killer cell mediated cytotoxicity. It is the 47th row in my data frame. So we will use the GSEA plot function from the enrich plot package. So this is not from from our cluster profiler package, but they sort of work well together. We'll give it our GSEA result from cluster profiler. We'll tell it what gene set or signature we want, and we will ask it to produce a GSEA plot of that without a p-value table, and to just put the description or ID column as the label. So let's go ahead and run this and see what we get. And this could be a static plot. There we go. So I actually think in a lot of ways these look better than what we get off of the Broad Institute's GSEA software. I see my rank ordered list here. I see the genes for this particular signature and where each gene fell as it marched down that rank ordered list of my data. And here's the running sum statistic. I've told it not to plot a p-value, but I can change that to true. It just can sometimes muddy the plot a little bit and get in the way. But it'll now replot it with the p-value listed if I ask it to. There you go. P-value, it's a little bit off the screen, but you get the idea. So we can do that, and one thing I really like about GSEA plot is I'm not by any means restricted to one pathway. I can grab a few of these. In fact, if I really want to convince you that cytotoxicity is upregulated here, I can plot multiple enrichment plots on top of each other. And it's something I used to do manually using third-party software that now I can just do directly in R. So I'm going to pick these first three, and I just supply those to GSEA plot as a vector, and it will produce an overlay. So we can go ahead and run this so that you can see what I'm talking about. All right, so there you go, our three different cytotoxicity related profiles. Each signature has a different set of genes. You can see that the T cytotoxic pathway from BioCarta has very few, but they were all found at the top of my rank ordered list. So it has the highest enrichment score, and on, right? So it's a very compelling, convenient way to put multiple enrichment signatures into a small real estate space for saving you precious space in your figures. All right. Other great visualizations, especially when we're dealing with a lot more pathways than we can show like this, is a dot plot. It's a special kind of dot plot that sometimes you'll hear referred to as a bubble chart, or I'll often call it that. But as far as ggplot's concerned, it's just a geome point where you've mapped different aesthetics or aspects of your data to point size, point shape, point position, etc. So let's take a look how we can do this. But before we do that, we actually want to add a column to our data frame from the GSEA result that will be called phenotype. And when the normalized enrichment score of a pathway, let's take a look at recall what we're dealing with here. When the normalized enrichment score is greater than 0, so in other words, a positive enrichment, a curve that goes up, that's a pathway that by default is enriched in patients with disease. When the normalized enrichment score is less than 0, that's one that's going to be enriched in the healthy controls. So I want this new phenotype column to say disease when the NES column is positive, and I want it to say healthy when the NES column is negative. So that's another use case, as I showed you in the last script, for this mutate case when operation. So we'll run this and we'll take a look at what we've created. I'll try to give it some more space so we can actually see it. It'll display in just a second, and there it is, phenotype, disease, 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 and then healthy. All right, so now that we have that, this information becomes really useful in terms of laying out our bubble chart. I'm gonna now for illustration, take the top 20 pathways in my GSEA result, and I want the x-axis to reflect that phenotype column, healthy and disease, and I want the y-axis to be the labels of these signatures. It's then just a geometric point. I'll go ahead and plot this. While it's plotting, we can talk through it. Just giving enough real estate space for this to display. It's then just a geome point with the size of the point set to the number of genes in a given pathway, the color, set to indicate the normalized enrichment score, and the alpha, or transparency, of the data point to represent the p-value. All right. I also asked it to scale the color from blue to red. So we end up with this very simple way to show a GSEA result where we can see all the pathways enriched in disease. Again, normalized enrichment score being shown by color, so positive red, negative blue. And the size represents the number of genes in that signature. And they're all highly significant, so there's not a lot of value in the alpha parameter here. All right, now that we've done that, we have a couple great ways to look at our data visualization, and we're ready to move on to a couple more GSEA approaches, but we're going to save those for the next video.